And what is function in the judgment?
Okay. It involves in the organization of cytoskeleton, cell migration, cell division, and metabolic process.
What do you mean by that?
Okay, so it because when cyst enters the body and it transforms to trophozoite, the trophozoite will attach to the to the intestine, lining of the intestine. So this, with many other proteins, will contribute for this attachment. It is considered a part of its cytoskeleton. I have put the picture of the structure of the yardi, but then I raised it so I don't take more than 10 minutes. But it's contributing to its cytoskeleton that will help it attach to the lining. And also, as I said, that when cyst transforms to trophozoite, metabolically it will become active. As a cyst, it's not active. Then this also contributes in its metabolism.
35 minutes now. So, I have a lot more questions, but
You can save that.
thank you.
You're welcome.
Thank you, Annie. So we should protect the
عاد زيادة؟
اوكي لا لا
أنت حتزود؟
لا
شو تاخذ عادي ما؟
اوكي بس تعالي
تفتحها بلابتوب ولا شو؟
لا حنين بس تعالي ديو
شير سكرين حاول
what is
Please help yourself to
okay. Okay. Hello everyone. I'm Ra'ad Habib Al-Asadi. I'm talking about the challenges avoid detect infection disease current method used trace the problem COVID-19 example. The COVID-19 infection disease caused by SARS-CoV-2. The most people infection, but not feel some symptoms because the infection is very slow or not have more the symptoms. The patient not need go to the hospital, not need the treatment. This patient is very dangerous because the spread the disease transmission other patients. This COVID, this method help the spread of the disease other patient. The COVID-19 is very fast transmission between patient to patient because the disease the transmission by the other methods touching inhalation and water and anything can transmission other patient. This very defective. The challenges infection disease detect viral pathogen specific challenges. Is one of the very challenges. The symptoms, the COVID virus, same the other disease. I'm working in hospital. All days I seen some patient. All patient tell me I'm asleep under or condition is the cold, not COVID-19, because the COVID-19 the same symptoms other with the infection. either with the infection, influenza, or common cold. Rapid envelope. The cost envelope infection agenus complement detect. The challenges after this, the symptoms sharing the COVID-19 with other disease, the influenza or common cold. The challenges is very different because the symptoms same with the other disease, fever, cough, headache, muscular pain, or other symptoms the sharing with the other disease. The detect infection disease, expert life by COVID-19 person challenges does too. This reagent help the COVID-19 transmission is very speed, is very fast between patient because the COVID-19 transmission by other methods, different other patient, different the other disease. The COVID-19 same share the disease influenza or the common cold. Rapid transmission, asymptomatic carry pathogen envelope. Suggest solution. Carry methods include PCR test, serological test, and advanced technological such as SEBRA basic diagnosis. Has many disadvantages which make detect sure I prefer a transmission spread faster. Timely resort a global calculation, a new test. We spread a vaccine complex play carry a role tact these challenges. COVID-19 rapid test disadvantage. This one of the problem can use the rapid test diagnosis of the COVID-19 because some person the negative test, the rapid test, sure the person felt sure I not have the infection, I not have the transmission disease other patient. This is not correct because the rapid test is the rapid test is not correct after the made the antibody. The antigen detect test can be used in the demonstration of the able immunity reaction to SARS-CoV-2, including reaction pro infection of the vaccine. However, the analysis considered incorrect because it relates on antibody that releases after releases for 14 days after the infection, and because this antibody are same this screening in the case influenza or common cold. More accurate, a specific synthesis and rapid test required. PCR disadvantage. PCR test disadvantage. Disadvantage PCR, the disadvantage of the PCR technology in analysis virus. Like the COVID virus, there are also some disadvantage. Potential contamination. This one of a problem can make analysis PCR assay. Contamination assessment. The contamination assessment is one of the problem the PCR because the contaminated during collection the sample or a preparation leading in correct result. Optimal cost. The branch and the minting cost requirement in chemical reagent of the PCR can be relative high. Time contribution. The PCR need more time, long time after take the result. Some patient waiting 1 days or other 2 days after they take the result. This patient can you transmission disease after can you transmission disease other patient? This disadvantage PCR because patient waiting result. Suggestion made on the technology detect the COVID-19 enveloped artificial interlink to of covered virus COVID-19 rapid screen. Mounting the extent complement with the prevent measure and the maintaining evolution. Development vaccine find more the acquired and fast technology and less expensive. Thank you. Brad.
Thank you, Rahan, for the presentation. Because it's topic COVID-19, I'm sure you are medical doctor. Are you what? What your position? Or what your
Technician medical.
technician medical? Okay. Are you dealing with COVID-19 screening before in your country?
Yeah.
Okay. I'm not sure on the last slide. Can you go back to the last slide? This one. Actually, I'm not sure how. I mean, for the previous one, the disadvantage kind of things, because you discussed about the advantage, and it's probably the best actually you compare other method and by create a table comparison, so you can see clearly what the lack of disadvantage was the PCR. You can by using table is more easy to compare like this. You put on the text, but people could compare what the reasonable for it. And this one for suggestion, I think suggestion, I'm not sure. For the third one, developing vaccine, I think everyone know you have your vaccine already, right?
Doctor, the artificial, this application used in Emirate Airflight. This specific diagnosis any patient have some symptoms. Not need the L patient check or the take a specimen collection. This need this use more time. This a fast diagnosis patient. Not expensive, not.
No, I mean the third one, not the first one. You mentioned the one. I mean the vaccine is developed. I think.
Yeah. Now there's some problem in vaccine. I reading some news found the other country use some problem in vaccine. British and France because the virus have some mutation, not a specific vaccine. Now, can you make the development vaccine prevent the infection virus? Okay.
What you mean is, you mean you want to develop some sort of vaccine that's specific for different type of mutant. Every time the mutant come out, you develop vaccine. Every time mutant come out, you develop. So how many vaccine you gonna have? One person.
Victor, because the made of the vaccine now is very fast. Not have the more time the student about of the vaccine L country because the more infection speed infection the L country need the vaccine. Some company research about of the vaccine can make any vaccine. You help stop the infection. You help some patient.
Okay, first one is artificial intelligence. What's mean by it?
Yeah. We use artificial intelligence to overcome COVID-19.
As ChatGPT to.
Yeah. Now some country use artificial in diagnosis of diagnosis the COVID-19. This application use it in Emirati fly. This application not need the speak with the take the specimen any patient. Can you using the application screening the L area, diagnosis the patient have some symptoms sharing the infection COVID-19.
So, yeah, I understand what you mean by artificial, but really,
Not confirming, right?
yeah, it's not completely.
Predicting.
Yeah, predicting.
You're
So
predicting
like you say, the first you mentioned when you got infected, it's very, how to say, spread very fast with one person. Then how you want to detect one person? You want, I think for Malaysia, I'm not sure lah. In Malaysia,
Yeah, found in Malaysia.
Malaysia way just isolated you fasting, isolated, keep you isolated and
Quarantine, yeah.
quarantine you. You cannot go out. I'm not sure in the country, but maybe artificial agent, yes. But if you
Can development to the artificial, not to use it now.
maybe
Yeah.
artificial, maybe you can do a simulation like one person stay, even get infected that area, then the whole village must be quarantined because of this area. Maybe something like this. I'm not sure about the disease. And I think we quickly come out, I think, monitoring the extent of some compliance and community measure. It's how this surveillance side, something surveillance side. Monitoring the extent of compliance and preventive measurement and monitoring regulation. So it's come out with CCTV. I'm not sure everyone will like CCTV around your house to check you whether you go out or not. I think in some extent, even privacy is important also. Not everyone, if let's say you are a COVID patient, then you are.
Victor, the COVID-19 is a new infection. Need some development the methods can use the method diagnosis or a treatment. Now this under of the study, this under the development. Not sure about of any application, any vaccine because a new infection needs a student need the research about the COVID-19.
Okay, next last, find a more accurate and fast technology and less expensive. What's mean fast? Faster than current method? What's faster? What you can think about it?
Can you development the rapid test? The rapid test now need more time. Need between the 7 or 14. Yeah. No. Victor, the antibody made after the 14 days.
14 days.
Yeah.
Okay.
Yeah, the antibody. This not correct because the patient sure after the test, sure that I not have the infection.
Okay.
Because the antibody after the 14 days they made.
Yeah, but the most of the rapid tests are virus detection, not
Cheap already. How cheap can be? Yeah, I don't know how cheap can be.
few ringgit, are they?
Yes, you buy it and more.
No. Development. Another the rapid test. Not now the rapid test. The rapid test now.
Information accurate and fast. I think fast is. I'm not sure how fast can be because current method although it's not the best, but everyone can purchase.
The antibody made after the 14 days. This is a problem.
14 days is very different. Even after you get infection, you quarantine for 5 to 7 days.
Sick already.
Already gone already, right? One thing I ask, have you been?
Victor, I'm working in hospital.
I know.
Some patient found some symptoms after the check by the rapid test not have infection.
Yes.
This is a problem because they made the antibody after the 14 days. This is a problem. Can make now I ask can make the rapid test fast and detect a new infection. Development.
Not sure because I don't know how fast can be, how cheap it can be. Less expensive, how cheap, I'm not sure. Now it's only about 40.
After the development,
I
not
don't know,
expensive.
maybe the cheapest way you can just put
Yeah.
piece of paper, just piece of paper and come up, that's cheaper lah.
For your tongue and
Yeah, just put paper come
Victor,
out this.
I'm sure the rapid test is not correct now because this. Yeah.
One thing is, I'm not.
No.
Because, one thing I have to correct your manner of presentation. Don't when people speak, don't interrupt too much, no. It's annoying other people actually. It's counter. I know you want to defend, but because I'm now questioning something. So at least you give me the
Sparse.
time to. Yes, I understand the period of 14 days or this kind of thing, but when you are sick, the way is quickly to isolate the thing with first person. Have you been diagnosed with the COVID-19 before?
Yeah.
How many times?
The symptom after this,
Everything.
The-
Confirm guess.
after the 4 days, 5 days,
How many times you gonna? How many times you?
three. L infection, not same the symptoms.
Spindle.
Different because the COVID-19 have the some mutation. Not
Yes, I understand.
yes.
Everything got mutation. Every now got strain already. You cannot just in the system healthcare, in the public healthcare, we cannot say you got COVID-19, got alpha B, one day one. Nobody care. But when
Chromatin.
the people dying is the best. Now you discuss the situation is the best already, right? Everything got vaccine, everyone had booster, everyone in Malaysia we got booster waiting. So it's good situation. In the last time, remember when you got
Victor, now the
Tumors.
specific COVID different and other COVID because the COVID have the mutation.
But I think for your word presentation is okay. But I think you can
Necrosis.
do more. I think improve a bit more. I think your presentation, I think not that confident now. Maybe thank you very much.
Thank you, Victor.
Thank you, Dr. Thank you.
Thank you.
So we can move next. Now,
هذا تعلمون
6,000 genetic disorders have been known. Lack of effective treatment strategies made the scientists to think about something like helping with the treating the underlying cause of the genetic disorder, not only the managing the symptoms and controlling the disease. So they came to the conclusion like CRISPR-Cas19 can help with that thing with the by offering the innovative therapeutic solution. So what's CRISPR-Cas9 system? So we know like CRISPR came from the short is from clusteral regularly interspaced short palindromic repeats. It's a technology research scientists use to selectively modify or dealing with the DNA of living organisms. It came from the adaptive immune system in the bacteria and archaea. They found it like this one. It's working, helping the bacteria to be immunized against any nucleic acid entering the bacterial cell. So they found like in 2012 they could develop the CRISPR-Cas19 CRISPR in the lab and help it to modify the genetic sequence, DNA sequence within the lab. So in that finding, helped the scientists to use the CRISPR till now for different in different fields, versatility of the CRISPR-Cas9. So what's the component of the CRISPR-Cas9? CRISPR-Cas9 system composed of the two-part Cas protein, like working with the molecular scissors, cutting the double-stranded DNA with the help of the guided RNA that complementary bind to the targeted sequence. By the target specifically and precisely binding to the target sequence, made it like to be precisely cutting the DNA that we want. And this is all about the principle of the CRISPR-Cas9. What about gene editing of CRISPR-Cas9? It came with the help of the repair system within the human cells or the living cells. By the help of the DNA repair system, we have two repair systems, non-homologous engineering and also homologous directed repair gene. This all belong to the knock-in of the gene and also knock-out of the gene. With the help of these two mechanisms, the DNA repair helped the CRISPR-Cas9 became a genetic tool nowadays in the 21st century. So About my presentation or the research that I choose to do, my problem statement is about how could CRISPR-Cas9 basic therapies potentially replace lifelong medicines as the primary method for controlling genetic disease in the future. So till December, the CRISPR-Cas9 was in the labs and clinical trial didn't approve it like this one is going to be therapeutic purpose or not. What about the research question? How CRISPR-Cas9 could system to avoid unintended genomic alteration, because CRISPR-Cas9 will deal with the genetic information. As we know, like genetic information has the effect of our phenotypic and everything's outcome of the genetic. So how the CRISPR-Cas9 or a researcher can avoid unintended alteration of the genetic and also adverse effect on the targeted cells. After we did the editing the gene, how the cells be avoided from the adverse effect. So, firstly, like the milestone and the dream came true after the FDA approved the Cas9, first CRISPR therapy for sickle cell disease, sickle cell diseases. So Cas9 is a cell-based CRISPR edited therapy utilizing CRISPR-Cas9 for editing the genetic within the cell-based gene therapy is approved for the treatment of sickle cell disease in patients 12 years of age and older with recurrent vaso-occlusive crisis with the sickle cell crisis. Patient hematopoietic blood cells, stem cells, are modified by the genome editing using CRISPR-Cas technology. So this Cas9 came from the CRISPR Therapeutic and Vertex pharmaceutical company, like being approved and announced in the 8th of December 2023. As the scientists that have found the CRISPR-Cas9 in the lab, she mentioned that in going from the lab to an approved CRISPR therapy in just 11 years, just a truly remarkable achievement and opening the door for different genetic disorder to be addressed and solve the problem, long life management of the treatment for them. So why sickle cell disease? Why they worked on the sickle cell disease? Because sickle cell disease has effect of the socially and economically on the USA. Because every annual birth, like worldwide, annual birth like 300,000 baby born with the sickle cell disease because of the African American family in America, and also why there's mentioning the beta thalassemia. I will mention that thing. Sickle cell disease is a congenital hemolytic anemia caused by an inherited point mutation in the beta globin gene on chromosome 11. As I mentioned, inherited point mutation. This point mutation helped scientists to think about CRISPR can solve that thing, because CRISPR cutting the genes, if there's single mutated gene, so it's easily can be edited and cut it or knock out or knock in that genes. So why they mentioned beta thalassemia alongside with the sickle cell disease, because beta thalassemia also suffering this phenotype of the disease comes from the mutation beta globin genes. Treatment option for the sickle cell disease only limited for the hematopoietic stem cell transplantation, and this one it's limited. Why? Because they didn't have enough donors, or some of the patients didn't have donor for transplantation. And rejection of the engraft, one of the causes that didn't let this treatment to be successful for sickle cell disease. So how Cas9 working? The principle of Cas9 it's not about editing beta globulin. So CRISPR-Cas9 targeted the effectively inactivated BCL11A repressor transcriptional repressor factor. What is that repressor factor? As a fetal, when the baby born, we have fetal HbF, fetal hemoglobin. This fetal hemoglobin after the birth will change to the adult hemoglobin. It's going to be beta globin, from gamma globulin to beta globulin.
So in that case, like decreasing the fetal hemoglobin and leading to the beta globin or adult hemoglobin to rise. And with if the baby, the one sickle cell affected patient, will increase with the mutated genes of hemoglobin, so it will face to the misshaping of the crease shaping of the red blood cells. So the scientists thought about that. Instead of the changing the beta globulin, cutting that gene, they found it like BCL11A, a repressor factor that repressing fetal hemoglobin, and they found that the baby that born till 3 months there is no any sign and symptom for sickle cell disease. So they found that because of the presence of the hemoglobin, LPF hemoglobin, so they worked on this approach and they developed this CRISPR-Cas9 to attack BCL11A, a repressor factor of transcriptional of gamma globulin. After inhibiting, CRISPR-Cas9 with the Cas9 cut it, knock out the gene BCL11A from the hematopoietic stem cells of the patients. So after they knocked out this gene, so there is no transcriptional inhibiting, there is no transcriptional factor to inhibit gamma globulin to be expressed. After that thing, sorry, I didn't mention that thing. This CASGV treatment, it's based from the stem cells from the patient own cells without, it's autologous. So after collecting the hematopoietic stem cells and blood cells treated with the isolated only CD3 or T-based cells and transfixion and electroporation with the Cas9, CRISPR-Cas9, that attack the BCL11A, and after editing this gene, cutting or knocking out this gene, so after knocking out this gene, so they found it that the hematopoietic stem cell edited one can express the gamma globulin, and the gamma globulin will help the sickle cell disease to live a normal life with the hemoglobin that's coming from the fetal hemoglobin. Sorry. Okay. What's the effectiveness of CASGB? CASGB, the safety and effectiveness of CASGB were evaluated in ongoing single-arm multi-center trial in adult and adolescent patients with a sickle cell disease in clinical trial. The therapeutic goal is for patients to experience fewer vaso-occlusive crises in sickle cell diseases. The primary efficacy outcome was freedom from severe vaso-occlusive crisis episodes for at least 12 consecutive months during 24 months follow-up period in clinical trial before approving by FDA. So a total of 45 patients were treated with the CASGB. 29 of them achieved this outcome without, they live it without blood transfusion and also visiting the hospital. All treated patients achieved successful engraftment because it's autosomal from its own body cells. Patients who received CASGB will be followed in long-term study to evaluate long-term its product safety and effectiveness, because they evaluated for 2 months and they cannot wait for long period and what happened within the human body. So they will evaluate long-term within the body for they say it's about 12 years. What's the advantage and advancing in the CRISPR-Cas9 helped scientists to approve new therapeutic from this system? First, efficient and precise of the CRISPR-Cas9, that precisely editing the specific nucleic acid sequence, making it a powerful tool for the genome editing. Convenient and programmable, because the CRISPR-Cas9, we don't have to program to engineer the proteins because it's challengeable before the CRISPR-Cas9. Only the thing that the scientists will do, designing the specific single-guided RNA to target the desired DNA sequences. Cost-effective, the CRISPR-Cas9 system, as used the system in the lab, it's cost-effective. Why? Because there is no need for the protein engineering or something like that, due to its simplified design and the availability of open source online software for guided RNA design and off-target prediction. Versatility of this application, CRISPR-Cas9 can be used in the many different fields, a range of application. Enhances precision technology advancement help lead to the development of CRISPR-Cas9 derivative with the enhanced precision, reducing off-target effect and improving the safety and efficacy of the gene editing that led to the approval by the FDA at the last HGV treatment. Off-target detection, high-throughput sequencing and computational algorithm analyze possible off-target location and optimize guide RNA design to discover and limit off-target effect. Because the only concern about the CRISPR-Cas9, it was the off-target effect after the treatment. Strategies to reduce off-target effect in CRISPR-Cas9 genome editing. As I mentioned, it's all about improving the guide RNA design and modified Cas9 enzymes. Production of the Cas9 from the streptococcus genes, it has to be optimized and then use it in the patients. Cas9 protein engineering, if the Cas9, we can engineer it in better way. Deliver methods. Choosing appropriate delivery method, because till now the CRISPR-Cas9 treatment is ex vivo, not in vivo. Like they cannot use it inside the human body because of the any off-target of the CRISPR-Cas9 and also the immunological responses. Validation and screening after the after treating patient with the cell editing with the CRISPR-Cas9, it has to be evaluated and screened for the sequence, what happened after the editing of the genes. Continuous research and development. Ongoing research efforts are focused on improving the specificity and accuracy of the CRISPR-Cas9 to further reduce the off-target effects. Ethical consideration as the CRISPR-Cas9 for sickle cell disease anemia, it's not about germline, so there is no any ethical consideration about the prohibiting this system. Only it's about accessibility to all humanity and affordability for everyone. The cost of the CRISPR-Cas9 treatment. My conclusion is all about the CRISPR-Cas9 is helped with the futures addressing the genetic disorders to be treated and also manage it to better outcome of the patient's life expectancy and their lifestyles. This is my reference, and so thank you so much.
Thank you, Tana, for your presentation. Dr. Daru.
Yes, yeah. Thank you for the very comprehensive presentation. I have a general comment first before I go for scientific comment. General comment, you don't have a slide number.
Yes, I
You
didn't
have to put slide number.
Okay,
So
sorry.
easy for the people to ask question, by referring to the slide number. And then
Oh, sorry.
Yeah. Another thing, I think you're too fast. Maybe you can slow down a bit. And when you speak, sometimes you don't really refer to the slide. You have something in your mind come out, and you are talking too much and too fast, and then sometimes it's a bit difficult to grasp. So sometimes technical presentation, technical facts, probably you can have certain diagram, like it can be a quite new things for your friends. You have to always remember when you're presenting to your friends. You want them to understand what you present. Sometimes certain things may be new to them, like not in and not out. What is not in, what is not out? You mentioned that. So probably you can have a bit of diagram. This is what you mean by not in, this is by mean by not out, So certain technical thing, you can present it with a diagram will be quite helpful for others to understand. Okay, okay, my other question. You have used this, can you go to the
Which one? Sorry.
that slide.
This one.
Have you, the previous slide also.
Cas9. Okay. I don't know why it's not working good. This one.
Yes.
KGV.
Yeah.
Okay.
So have you come across other, why you choose this for your presentation?
Because I was too much interested in CRISPR-Cas9, because it's related to the bacterial adaptive immune system, and we can use it for the life, in the medicine, like precision medicine. So secondly, when with the like I have news like articles. So I saw, oh my God, KGB, they approved it, like there is a treatment for, they came like there's a result for the CRISPR, because it was all, they said like it's all in lab. So when I saw like KGB, it's treatment for this, and they approve it, and it's will use it. So I'm just interested in that thing.
So far, how many CRISPR treatment have been approved?
Yeah, this one is the first one, like being approved to use in the human cells, humans, inside the human. This is the first one, and they are hoping for coming new, but like overall like 71 clinical trials are going in the under the CRISPR-Cas9 for the treatment for belong to the cancer, belong to the sickle cell disease, and also like immunotherapy. But all of them, what I like during my preparing my presentation, I was like thinking CRISPR-Cas9 like it will be given to the patients inside the human body. I was thinking like that, but then I realized like it's all cell-based gene therapy.
Okay, so far, do you think what is the biggest challenges for them to approve CRISPR-Cas9 as a gene therapy?
So,
Why so far only one that has been approved? What are the drawbacks or major challenges for other treatment that is not being approved or in queue?
belong to the CRISPR-Cas9.
Yeah.
So
Why so far only one?
this all of them, like all the cancer and broadcast or CRISPR-Cas9, it's off-target thing. But about the KGB, I found it like I didn't want to even mention the like challenges and how to overcome the off-target. Why? Because from KGB they use it all the after all the research they choose it all the like how the overcoming plan for the off-targeting. One of the things like they use it here I mentioned it like RNP. Sorry, from this one RMP, they bind it because all the others like using Cas9 alone, like inserting the Cas9 alone, and then guided RNA, and then the other factor for making them together inside the cells. They found it in that case like the CRISPR Cas9 will attach to the Cas9. It's not combined to the guided RNA. So one of them they use it I forget this one, RNP, ribonuclease proteins, like bind it together, and they, like minimize it, the off-targeting by this one. Secondly, like they use it, what else they use it? I forget it. So they use it all the,
What's the delivery method they use?
viral. Yeah, there's a concern about like this viral maybe have side effect about the genetic changing inside the cells. And what else like concern they have found it the delivery one, and also off targeting, but they the other thing like they minimize the knockout thing. They use it knockout in the KGB. Why? Because knockout there is no insertion of the gene inside the sequence, so there is no new like shifting of the sequences. So they say like they are saying like as much as the researcher or the system they can provide with the like going with the homology directed repair, it minimizes the off target thing, minimizing the off target. And also like minimizing the off-target thing, other thing like from the KGB, they use it the company software like bioinformatic tools for like checking the sequencing. So this this sequence of the guided RNA and Cas protein will bind to which part of the whole sequences. So they minimize it to 1 to 3. After they came for minimizing 1 to 3, so then they use this Cas protein for the KGB.
Okay, I think you read.
I read it a lot, but the problem with me,
because you can answer quite well. Okay,
the problem like for knockout and knock-in, I put in the slide about this problem, but it was too much. Like I couldn't minimize all.
Yeah, it's okay. Thank you.
Okay,
That's all from me.
Thank you, Dr.
Sorry, like I think I took a lot of time, but because of the wide range of the things I wanted to do it, so I couldn't, like practice and minimizing everything.
That's why I think all of you need to understand, because the time is only 10 minutes.
Yeah. Yeah.
You have to really just focus on one thing maybe.
Yeah.
You can't do everything because it's not a lecture.
Yeah.
So if you have taken off, something, then you can concentrate and finish it.
Yeah.
So this may not be, you don't have to do this, but
Yeah.
you can concentrate on that.
Only I want to mention that thing right now after they approved like with this good news for the Vertex and pharmaceutical company. Right now they are working on the how to use CRISPR directly inside the human cells because with this one the patient had to go to before like receiving that edited gene have to undergo the chemotherapy and a lot of the 1 year of long treatment. So they are working on that things.
Thank you.
Thank you. Thank you so much.
Yeah.
I see that. Look up here.
Sorry.
Not too much, so close.
Share your statement.
Yes, yes, I want.
Your literature. There is not like that.
How to cancel this one? Sorry. Okay.
Yeah. Some can you share your tries and start your prediction?
Okay, sure.
Okay, so can you share your slide?
Yeah, yeah.
So please on your video and share your slide. Okay. Where oh yeah, it's received.
Hello everyone, my topic is the AMBRA1E3 ligase adapter regulates the stability of cyclin D. CDK4/6 is a key regulatory factor in the cell cycle. By forming a complex with cyclin D, it can phosphorylate the RB and release transcription factor E2F, promoting the transcription of cell cycle-related genes and allowing cells to enter the S phase. CDK4/6 inhibitors effectively block the progression of tumor cells from G1 phase to S phase. In EI-positive breast cancer, CDK4/6 will be overexpressed, resulting in uncontrolled cell proliferation. At present, CDK4/6 inhibitors in clinical practice can inhibit cell proliferation downstream of signaling pathways. Multiple CDK4/6 have entered the clinical stage and achieved good results. The resistance mechanism of CDK4/6 inhibitors can be divided into cell cycle-specific mechanisms, including RB1 gene detection, E2F amplification, CDK4 gene amplification, and so on. The non-specific mechanism of cell cycle include activation of FGFR and the PI3K-AKT mTOR signaling pathway. Currently, the main focus is on RB1 gene deletion and the PI3K-AKT mTOR signaling pathway. The use of CDK4/6 is constantly being promoted to other cancer species, making it increasingly important to address drug resistance issues. Therefore, Therefore, exploring new CDK4/6 resistant molecules from this perspective. Next, I will mainly share the research ideas of this article. After treatment with hepcyclic audio 937, it was found that it can affect cell growth and the phosphorate of key cell cycle gene RB. Subsequently, whole genome sequencing of CRISPR-Cas9 in U937 cells revealed the RB1 pathway. After sequencing through the sgRNA library and sequencing, it was found that AMBRA1 had the highest differential expression. This whole sgRNA library sequencing method for drug-resistant genes is worth learning from. Subsequently, the also verified the relationship between AMBRA1 and the cell growth and cycle arrest. After treatment with hepcyclic, it can be seen that AMBRA1 affects cell growth. The subsequent cycle results indicate that knocking out AMBRA1 leads to an increase in active cells, indicating that AMBRA1 can both affect cell growth and be associated with cycle arrest. That is to say, when using hepcyclic, knocking out AMBRA1 increases the number of cells entering the S-phase. It reduces the effect of hepcyclic, allowing cells to enter the S-phase. Knocking out of AMBRA1 can lead to resistance to CDK4/6 inhibitors. Subsequently, another CDK Subsequently, another CDK4/6 inhibitor and the 4 other cells were used to validate these results. Afterwards, considering the activity of RB and the cycling kings, it can be seen from the following that knocking out AMBRA1 increased the phosphorylation of RB. Previously, in vitro experiments were conducted to demonstrate that knocking out AMBRA1 can cause resistance to CDK4/6, but now it's being validated at the molecular level. Next, another further validate and obtain the levels of cyclin D increase on AMBRA1 loose through Western blot. It should be noted that the proteins or genes related to the cell cycle validated in this article are all related to the CDK4/6 inhibitor pathway. Therefore, it is possible to search for the targets affected by AMBRA1 from existing mechanisms and discovering the mechanism of drug resistance caused by AMBRA1 deficiency. Next, the author validated it by constructing AMBRA1 knocking out mouse in mouse. By observing the growth and developing status of mouse at a different time period after AMBRA1 knockout, simultaneously seen with cycling reverse increase in cyclin D levels in mouse embryos after AMBRA1 knockout, verified up to now and confirmed that AMBRA1 can directly affect cyclin D. Due to previous studies indicating that AMBRA1 can promote autophagy and inhibit mTOR activity at MYC, all of which may affect cell cycling progression and response to CD4/6 inhibition. Subsequently, the author validated autophagy based on these classical pathways and concludes that AMBRA1 does not affect cell cycle proteins through autophagy. This is actually equivalent to an exclusive experiment to exclude the influence of possible mechanisms. The author also excluded the effects of mTOR and MYC, PRK1 and Aurora kings on cell cycle proteins using this method. Due to the previous analysis not finding a clear target, the author shifted their focus once again to cyclin D1 for further analysis. Perhaps the cyclin D1, which is the highest expressed molecule, is also the mechanism of change. A mutant of cyclin D1 T2 T2A6. Phosphorylation was constructed while analyzing cyclin D1. It is a state of overexpression of cyclin D1, which can promote cell entry into the S phase and reduce sensitivity to low dose publicly. Afterwards, AMBRA1 knockout was performed to observe this phenomenon, and it was observed that there was a significant change in the number of S-phase cells after knockout. A hair cell supplement. In the process of promoting cell proliferation, CDK4/6 need to form complexes with cyclin D1 or trimers with cyclin D1, P27 to promote downstream gene RB phosphorylation levels, thereby causing a function of E2F1 transcription factors and ultimately promoting cell proliferation. Previously, it was confirmed that knocking out AMBRA1 can promote an increase in cyclin D1 expression. Therefore, further evidence is needed to define whether this increase will cause an increase in the expression levels of CD1, CDK4, and P27, thereby leading to a resistance to CDK4/6 inhibitors. Knocking out AMBRA1 using CDK4/6 inhibitors for specific expression patterns between cyclin D1, CDK2, and P27 was verified through an experiment. It was found that after knocking out AMBRA1, the expression of cyclin D1, P27, CDK4 trimer increased. Further explanation suggests that AMBRA1 promotes the expression of cyclin D1, leading to the formation of cyclin D1, CDK4/6, and the P27 complexes, ultimately resulting in the inaction of the CDK4/6 inhibitors. It had been proven earlier that AMBRA1 can regulate cyclin D. So which pathway is used to regulate cyclin D? First, consider the degradation of cyclin D during the cell cycle. After such X treatment, the degradation cyclin D1 in the control group and AMBRA1 knocked out group slowed down. On the right is a degradation curve of the cyclin family D1, D2, D3. Therefore, we can conclude that AMBRA1 can regulate the degradation of cyclin D. Afterwards, the tube pull down was performed to confirm that knocking down AMBRA1 can display a lower level of nucleation of cyclin D1. The MS analysis was performed and it was found that the nucleation of cyclin D1 was a decrease in nucleation at the K33, K46, and the K238 sites. That is to say, knocking down AMBRA1 may result in changes in nucleation focusing on these 3 points. So far, it has been confirmed that AMBRA1 regulates the nucleation of cyclin D. So, how does AMBRA1 cause the nucleation of cyclin D? The idea is to find the lagging intermediate nucleation. Based on previous research, AMBRA1 can act through CL4 ring E3 nucleation ligands. So searching for the CL4 protein family is necessary. In the research process, the main approach is to verify whether AMBRA1 can continue to affect a level of cyclin D1 by altering the levels of CLB4 proteins. Afterwards, by mutating the AMBRA1 protein for replacement, it was found that it cannot compensate for the decrease in cyclin D1 caused by AMBRA1 knockout. AMBRA1 can cause changes in cyclin D1 through the CLRB4E3 ligands. Afterwards, the author conducted a validation on animals and clinical specimens. First analysis was conducted in the TCGA database, and it was found that 2% of lung cancer patients in the TCGA database had AMBRA1 level mutations. Therefore, the author speculates whether AMBR1 can act as the tumor suppressor to exert its effect. Afterwards, the author provided this hypothesis in a quiz-driven long decreasing mouse model. The author constructed 3 types of mouse through quiz9. In this model, AMBR1 is used as the target gene, and the other 3 are used as positive control tumor suppressor genes. After transcription of sgRNA into both KTC and KPTC cells, the survival ability of the cells was significantly enhanced after knocking out AMBAR1. However, there was no change in the KT mouse, further proving that AMBAR1 can play a role in initial cancer. Finally, the author validated the prognostic relation between AMBAR1 and the lung adenocarcinoma using a backlink from lung adenocarcinoma and then analyzed it in a public database. Further evidence has shown that AMBAR1 can have an anti-cancer effect. However, there's also a problem here. The purpose of the study is to investigate the drug resistance of AMBAR1 in CDK4/6 inhibitors. In the final clinical and animal experiment, only its anti-cancer effect was studied, and the interaction with CDK4/6 inhibitors was not studied. Moreover, the conclusion that AMBAR1 is an independent risk factor for CRISG mutation in lung adenocarcinoma can only be seen in patients with CRISG12 mutation. Therefore, the author concludes that AMBAR1 is the tumor suppressor factor in CRISG12 mutation lung adenocarcinoma patients. This is the basic ideas of this article. This article provides a good research approach in studying the mechanism of drug resistance. The mechanism argument is very rigorous, using CRISPR-Cas9 for whole gene sequencing and the mouse model construction, as well as constructing multiple mutants with relative high technology barriers. A new tumor suppressor factor AMBAR1 has been discovered, which may serve as a new drug target for CDK4/6 inhibitor resistance. But there also have some disadvantages, such as both the beginning and the end of the article mention the main use of CDK4/6 inhibitors for ER-positive patients, but only one presentation of MCF7 data is presented in the article. Furthermore, since AMBAR1 may serve as a site for resistance to CDK4/6 inhibitors, another experiment should be conducted to determine whether the addition of CDK4/6 inhibitors can reverse its resistance. This can conduct further research on this point. In addition, I think it would be better if we could make a breast cancer model in the animal experiment part. Thank you. That's all my presentation.
Okay, thank you.
All right, thank you, Sami. Presentation, can you hear me?
Oh, okay, I can hear you.
Okay, all right. First of all, I think what the title that you gave to Dr. Sasi is different from the title that you presented today, because I accept this to be the examiner because I thought it would be a general discussion on drug mechanism on cancer tumor, but it seems like you choose a topic that very specific on mechanism of the AMBAR gene, and then and it seems like you are just reading from text. I don't know, maybe you have text for your presentation. So in your own words, do you understand what is the work that you presented today?
Again?
in your own words, can you summarize what is the objective of this work and what is the final findings that when they knock out the ANBRA1? Can you summarize the work that you present today?
Okay. As in this article, the author meaning found that the AMBRA1 can
How does this knockout of ANBRA1 affect the cell cycle?
knock out. It use
Okay, in this slide,
it use CRISPR-Cas9 and find the RB1 pathway. Then use sgRNA library to
did they mention which mechanism, AKT or P13 cell cycle? I think the most appropriate way to present is when you choose a study that you want to present and discuss, you should have a proper introduction because you might, your audience might have different background. Not everyone understand what is about cancer. So when you put this background as mechanism and then you don't have a proper introduction, I think that would be the biggest mistake, I think, because everyone that difficult to understand the whole study if you didn't introduce well about the work, your objective, and then what you presented today is most of the findings, the results, which I think if you can understand, maybe it's best if you can explain in your own words. So I just want to know what is the final the most important findings that they found from this work.
what the most important finding in this article.
Sorry?
The most important finding in this article, new tumor spread factor ANBRA1 being discovered, which can
This cycle one.
the ANBRA1 can also new drug target for CDK4/6 inhibitor resistance.
Okay lah. All right, thank you.
Okay, thank you, Song, for your presentation. Now, thank you, Dr. Saida, for the comment. And now we move to next presenter. Okay, your evaluator is Dr. Anis.
Good morning, everyone. Good afternoon already, sorry. So today, we have already been through ups and downs in COVID-19, and it's been 4 years since COVID-19 first outbreak in Wuhan, China. So today, my topic will be related to COVID-19. and I'm Ian. And let's look at some figures before we dwell into the topics that I'll be sharing today. Okay, so there are currently 8.1 billion of total world population on Earth. Okay, and 700 million out of 8.1 billion people actually had COVID infection before, and 1% of the people actually died because of COVID infection. And this 1%, it looks like it's little people, right? But it's actually up to 7 million of people. So when we look at this figure, the ranking here of epidemics and pandemics with at least 1 million of deaths, COVID-19 pandemic actually ranked number 5 out of all historical epidemics and pandemics. Okay, so it caused around 7 to 36.6 million of deaths as of December 2023, which caused total global population loss of 0.1 to 0.46%. So we are creating history here, guys. We are in the ranking, okay? So since COVID-19 is so serious and it can cause so many deaths, right? Is there a biomarker needed to detect the severity of COVID-19 infection? Everyone is like, okay, just detect COVID-19 and then you confirm you quarantine. But how to detect the severity of COVID-19 infection? Okay, so here this leads to my topic that I'll be sharing, which is D-dimer, which is to detect the severity and mortality of COVID-19 infection. Okay, in brief, what is D-dimer? D-dimer is a byproduct of blood clot and degradation, which is useful for detection of ongoing fibrinolysis. And formation of D-dimer forms when thrombin mediates the cleavage of fibrinogen to form a fibrin, in which this will lead to fibrinogen to expose the binding sites in which the protofibrils will be formed. And here the protofibrils is a fibrin polymer in an overlapping fashion. So it is an unstable fibrin mesh. It is an unstable fibrin mesh. And with factor 13A, which triggers the cross-linking of the adjacent fibrin monomers, it forms a stable fibrin mesh for blood clot formation. So D-dimer is formed when this blood clot of the fibrin mesh is degraded into a fibrinogen degradation product and D-dimer. So this is the product that we are looking today, D-dimer. So currently D-dimer is already in use in laboratory test settings, as they are used for detection of thromboembolic events such as disseminated intravascular coagulation, deep vein thrombosis, and pulmonary embolism. And it is quite specific as it is formed only within 2 hours of thromboembolic events, and it has a short half-life. Means that if you are having elevated D-dimer in your body, it means that you are having thromboembolic event in your body currently happening. Okay, so what is the association with D-dimer with COVID-19? Since COVID-19 is a respiratory disorder and D-dimer is like something like blood clotting disorder. So what are the association between D-dimer and COVID-19? First of all, many studies have already shown that many mechanisms are involved in the COVID-19 that cause coagulopathy, which leads to elevation of D-dimers. Let's go into the mechanisms one by one. Okay, so first of all, hyperinflammatory response caused by COVID-19 will lead to mononuclear cells to produce a lot of tissue factors, which will generate thrombin. This thrombin, as we remembered earlier in the slides, it will cause fibrinogen to convert to fibrin, in which it will form a platelet clot. And here, this fibrin will tag onto the receptors of the inflammatory cells, which will lead to more pro-inflammatory cytokines and chemokines to be produced during the infection. Hence, it will shut down the fibrinolysis and impair the natural anticoagulant pathway, which leads to increase in D-dimer production in your body. Okay, next is the endothelial cells dysfunction. As we know, COVID-19 actually infects our endothelials, our cells through angiotensin converting enzyme 2. So endothelial cells also have this receptor. In normal condition, endothelial cells usually helps in maintaining thromboresistant condition by secreting various protective substance. However, during COVID-19 infection, the excessive stimulation of endothelial cells by virus on the endothelium directly or through immune mediation cause endothelial cells to die through apoptosis, which leads to a procoagulant state, leading to increase in D-dimer formation in your body. Okay. Next is tissue hypoxia. We know that COVID-19 infection can cause capillary hyperperfusion, which leads to tissue hypoxia. So in our body, vascular response to hypoxia is controlled primarily by hypoxia-inducible transcription factor, which is a HIF, which have two subunits, which are a alpha and beta subunit. Okay, so in normal condition, alpha subunit will be catalyzed and degrade by proteasome. However, in hypoxic condition, the VHL here will be depleted due to hypoxic condition. And this alpha subunit will then bind to the beta subunit and go into the nucleus and transcribe all the factors that promote thrombosis, such as plasminogen activator inhibitor, PAI1 inhibitor of plasma fibrinolytic activity, which leads to fibrin deposition. Okay, and next is neutrophil extracellular traps, which is the NET. So NETosis is a regulated form of neutrophil programmed cell death. Okay, so this what happens is the neutrophil will extrude all the DNA, histones, and antimicrobial proteins, which looks like a web-like structure as shown in this figure, known as a neutrophil extracellular traps, NET. So in SARS-CoV-2 infection, it will cause production of a lot of reactive oxygen species, which will lead to NETosis of the human neutrophil. Then the extrusion of this DNA, histones, and all the internal organs of the neutrophil will lead to triggering of the thrombin generation by binding to factor 12 in the body, which will enhance the anticoagulant, the diminish of anticoagulant factor and poor fibrinolysis. So this is the main mechanisms that cause immunothrombosis that in the COVID-19 infected patients. So here are the summary again: systemic hyperinflammatory response, endothelial cells dysfunction, tissue hypoxia, and NETosis all together leads to elevation of D-dimer in the body. So is the D-dimer as it is so good, right? It's like, okay, it looks promising. Is it good enough for for D-dimer to be a biomarker? So many studies have found that severe disease can cause elevation of D-dimer, or D-dimer of more than 1 microgram was found to have higher risk factor and poorer prognosis for patients with COVID-19 infection. And apart from that, D-dimer with more than 2 microgram per ml also have a higher mortality rate as found in many studies as shown. So it looks like it's very promising biomarker. And what are the current guidelines that we are having now? So IFCC interim guidelines on biochemical hematological monitoring of COVID-19 patients recommends that patients with COVID-19 infection to test for D-dimer, which is to identify the risk for adverse outcome of COVID-19 infection. And Malaysia Guideline Clinical Management of Confirmed COVID-19 case in adult and pediatric in 2022 May has also recommended that children with stage 4 or 5 COVID-19 infection to be tested with D-dimer to as there is a concern of worsening of disease for their COVID-19 infection. Okay. However, although D-dimer looks as a very promising biomarker, it is very less been adopted in a routine laboratory assessment of COVID-19 infection. This is due to high baseline variety of D-dimer tests currently that all the studies shows. Here, the assay cutoff value of so many studies are also different. That ranges from 0.5 to up to 3.647. So the cutoff value for D-dimer is very hard to be determined. Which cutoff is very severe? Which cutoff is mortality? So due to this reason, it is not used as a biomarker yet. Okay, and other limitation is like when to test. Is it the first day of COVID-19 infection? Is it the last day? When should I test this D-dimer? It's still not answered yet in so many studies. And lastly, it's not specific enough. As we said, it can be tested in many different diseases such as disseminated intravascular coagulation, deep vein thrombosis, and pulmonary embolism. Is this that diseases cause the D-dimer to elevate, or is it COVID-19 infection that caused D-dimer to elevate? So this is also point to argue, okay, for the D-dimer to be a biomarker. However, D-dimer is directly associated in disease severity and morbidity in COVID-19 infection, and it can be a promising biomarker if all the questions have been addressed as earlier, and it facilitates a personalized and more efficient clinical management of disease in a timely manner. Okay, that's all. The reference are shown here, and we have been through COVID together, and now we are still fighting. Let's go. Thank you.
I think this is one of the best signs of the present timely testing. This is a good example. I think others should follow that. Now we open for
Okay.
questions.
Okay. Thank you so much. So let's type the things together. Okay. Thank you so much, Ian. So your presentation, general comments, your presentations are very clear. You have introduced your topic very well, and you have addressed a few research questions, your topics. Okay. When you say D-dimer, and you also mentioned that it is not specific to COVID-19, it can present to other conditions such as malignancy, pregnancy, and also post-operative condition and all that. So why do you think that D-dimer as a potential biomarker when it's actually not specific COVID-19, it also can be associated with other critical conditions? Why it can be a potential biomarker for COVID-19 despite being no specific?
Okay. To answer your question, I think I didn't include the slide here, but okay, to answer your question is because actually there is a lot of testing that can be done to assess the severity and mortality of COVID-19, which includes like platelet count, APTT, activated partial thromboplastin time, and all that. However, all of that testing are also very not specific. So D-dimer is, how to say, it's like counted as quite less people with this kind of problem with elevated D-dimer. So it's more promising. And there are people who tested interleukin 2, 6, and all that chemokines and these cytokines. But it is also not specific, as other diseases can also cause inflammation in the body and cause elevated of these cytokines in the body. So there are no promising biomarker, but if we have like one or two tests that can give a guide to clinical management of this patient, then I think D-dimer is quite a good marker.
So, are there specific populations where the D-dimer levels are particularly of poor outcome? That means we should not consider D-dimer as a potential biomarker for this population.
That currently that I've studied so many articles, I have not seen any of the articles saying,
From your view,
from my view,
that should come up with that guidelines that this D-dimer biomarker potentially used for this population and not suitable for this population, mainly for COVID-19.
okay, perhaps those people with already had the hypercoagulability diseases should not have tested for D-dimer, because else you will have very high D-dimer, and does it indicate the severity of COVID? It cannot be how to say, we cannot correlated.
When you say severity, right? So how do you define the severity? Is it from no infection to how to say from mild to chronic, or how? Severity that this diver can use as a diagnostic.
Okay, so you can look at this page here. Actually, they already categorize what stage are you in. Then they also have to, of course, not only looking at the D-dimer levels, but they are actually looking at the clinical side. How is the patient? How is the is there a lower oxygen concentration and all that? And then they actually found out that during when you have higher D dimer, you have worse X-ray of lung outcome, and then you have also more systemic inflammation, which the patients are usually bedridden. Yeah. So this is how they assess the severity of COVID-19 for D dimer to associate the D dimer.
So, can D-dimer used as a standalone assay or determining the disease severity?
Sorry, come again.
Can D-dimer used as a standalone biomarker to determine the severity of the COVID-19 infections?
Actually, no. You cannot use the D dimer as a standalone. You have to combine all the clinical outcomes of the patient from clinical sites, and with laboratory tests with the confirmed COVID-19, then you can you can assess the severity of the COVID-19. Yeah.
Like this now.
Yes.
Because when you have a combination test, the specificity and the sensitivity
Will increase. Yes.
So with that, you can know that the D-dimer can best paired with what other tests.
Yes.
With the 100% sensitivity and specificity.
Right.
Okay, so far I'm done. Okay, thank you.
Okay. Thank you.
Okay, so we can move to next. Sultana.
Good afternoon. Is my voice audible?
You can share your slide, then you can start your presentation.
Am I audible?
Okay, you can put in the presentation mode. Okay, you can start. Sorry.
Good afternoon, everyone. I'm Sultana, and I'm going to talk on small extracellular vesicles for pro-oxidant delivery for drug-sensitive and resistant cancer therapy. First of all, introduction. Cancer is a leading cause of death worldwide, and multi-drug resistance hinders successful treatment. The World Health Organization revealed a sobering statistic that it's the first and second leading cause of death in the individuals under 70 years of age in 112 countries out of 183 countries. Amidst the challenges in cancer treatment, the phenomena of multi-drug resistance stands out as a formidable obstacle. MDR significantly contributes to the failure of traditional chemotherapy, leading to over 90% mortality in patients with metastatic cancer. Now, multi-drug resistance is the principal mechanism by which many cancers develop resistance to chemotherapy. MDR often involves overexpression of ATP binding cassette, ABC transporters, particularly B glycoprotein, which pumps drugs out of the cancer cells. MDR mechanism. At the core of MDR lies the overexpression of adenosine triphosphate, that's the ABC binding cassette transporters, particularly the P glycoprotein, which functions as a drug efflux pump, crucial for expelling cytotoxic drugs from cancer cells. Now, in plus to this, additionally, mitochondria play a dual role generating energy and producing reactive oxygen species. But cancer cells, especially those with MDR, exhibit high reactive oxygen species level, contributing to drug resistance. The delicate balance of antioxidants, particularly L-gamma glutamyl L-cystinyl glycine, GSH, it's the reduced form of glutathione, plays a crucial role. Several studies have shown that GSH level are associated with drug resistance in cancer treatment. Overexpression of sorry. Okay. Overexpression of glutathione S transferases, particularly GSTP11, is often considered as a possible mechanism of tumor cell drug resistance. Okay. So previously, the authors worked on benzoyloxy dibenzyl carbonate, which is a GSH depleting pro-oxidant that can rapidly decline the GSH, that's a reduced form of glutathione, and elevate oxidative stress to a threshold level above which cancer cells cannot survive and eventually die. In vitro studies have shown that the B2C, that's the benzoyl oxydibenzyl carbonate, exhibits strong anti-cancer activity against various cancer cell lines, leading to mitochondrial disruption, which uh, which is through activation of procaspase 3 and PARP1 and cleavage of BCL2. Other in vivo studies have also demonstrated that intravenously injected B2C causes apoptotic cell death in tumors and significantly suppresses tumor growth. However, in drug-resistant cancer cells, the efflux pumps poses a challenge to the efficacy of B2C. Pumps are responsible for mediating MDR, a process that renders chemotherapeutic drugs ineffective in the internal organs of the organelles of the targeted cells. Therefore, the efficacy of B2C in resistant cells may be limited by the flex pump. Introduction to small extracellular vesicles. So these are natural cellular structures that are present inside the cells. These are 50 to 20 nm. They possess lipid bilayer structure that enable the encapsulation of both hydrophobic and hydrophilic drugs. Originating from cells, SEVs plays a crucial role in cell-to-cell communication and the regulation of biological processes. Distinctive in their membrane composition, reflecting the parent cell's lipid and protein profile, SEVs exhibit a high degree of tropism. There is a specificity to that particular cancer cell towards the cell of origin. This inherent specificity eliminates the need for surface modification for effective tumor targeting, making them a versatile tool in drug delivery. Advantages of SEVs. Beyond their targeting specific capabilities, SEVs offer several advantages. That is, biocompatibility, low immunogenicity, stability against enzyme in biological fluids, tenfold greater adherence to an internalization by cancer cells. Their natural origin allows them to bypass the efflux system, releasing drugs directly into the cytoplasm via endocytosis. Now, the mechanism of action. So, the authors combined B2C with the small extracellular vesicles from the cancer cell line OVCAR, that's the ovarian cancer cell line, and they injected that into the cancer, cancer animal model, and they have observed this that BSEV because they are taken from the cancer cell, it has the tropism to that particular cells, and it undergoes, it reaches inside of the cytoplasm without interacting with the P-glycoproteins, and it causes increase in the reactive oxygen species and decrease in GSH, which increases the reactive oxygen species level, but it causes the cell death. So it leads to apoptosis. Therapeutic potential, oxidative stress modulation. So BSEVs targeting cancer cells, reactive oxygen species surge, mitochondrial disruption, oxidative stress amplification. It also has drug resistance inhibition because it bypasses the efflux pump, precision delivery of B2C to the cytoplasm, ATP starvation, and efflux pump dysfunction, antitumor activity, and programmed cell death. Comparative advantages, it has biocompatibility, low immunogenicity, enhanced adherence, internalization, minimal toxicity, and selective action. So the disadvantages of this process, small extracellular vesicles, is that it has limited payload capacity and complex manufacturing process, immunogenicity concerns, selective uptake by cancer cells, risk of unintended consequences, regulatory approval challenges, cost of production, and long-term safety concerns. The key findings are tumor derived from cancer cells. Tumor Small extracellular vesicles derived from tumor loaded with B2C present an innovative solution. VSCVs demonstrate therapeutic potential as oxidative stress modifiers and drug resistant inhibitors. presenting a transformative approach to cancer treatment. Despite these potential disadvantages, ongoing research and development efforts may address and mitigate these challenges, ultimately advancing the potential of BSEVs as a transformative modality in cancer therapy. Implications: further exploration of BSEVs in preclinical and clinical settings could lead to innovative and targeted cancer therapies, revolutionizing the field of oncology. These are the references. Thank you.
Okay, thank you, Sultana. So now I open for questions and comment from Dr.
Thank you, Dr. Sasi. Thank you, Sultana.
Thank you.
Thank you. All right. For these small extracellular vesicles, apart from what you have introduced for specific cases, could you please explain how expanded is this technology used at current stage?
This like have read that many researchers have used other drugs in place of B2C, but it just depends the target that they are finding. Like it may not be the reactive oxygen species that they are targeting, but it would be another mechanism that they would target.
All right. Talking about mechanisms, what is the mechanisms to produce these packaged small vesicles?
This like the process was a really complex process. Like it was beyond like I can comprehend. It was very tedious, very complex process. So I did not dwell into it much. Yeah.
All right. Um, in your slide, you show that the vesicle, you show an example that the vesicle comes from cultured cells.
Yeah, yeah.
Do you have any idea how this was done?
From the cultured cells.
Yes, from cells to the vesicle.
Oh, yeah. I have read about this, but I'm not. What was it?
All right, it's okay. Apart from a direct injection to the proximity of the tumor, if
Yeah.
you if we want to make a therapy, what are other mechanisms that you can think of for the delivery
For the delivery. But
of the
I have
vesicle?
seen. Yeah. So I have seen the site delivery, direct delivery, and not the um like intradermal. No, I think it's um in yeah. I'm not sure about this.
Okay, it's okay. That's all from me. Thank you. Okay,
Thank you,
Thank you.
Dr. So thank you, Sultana. So now we move to next presenter.
Thank you.
Thank you. Yeah, Puspa, are you there?
Yes, Doctor.
So, Puspa, you can on your video and share your slide. Okay, you can share your slides and start your presentation. Good
Good afternoon, everyone. Am I audible?
Yes, yes. You can start your presentation.
Good afternoon, everyone. I am Puspa, and the title of my presentation is Alternative Therapeutic Approaches to Conventional Antibiotics. Introduction, antimicrobial agents. We all know antimicrobial agents are the primary treatment for infectious diseases. They work at killing the pathogen, or like they inhibit certain functions of the microbes by binding to the specific structures. They have revolutionized the field of medicine by combating the microbial infection, and also they have transformed medical practices. Like they have enabled us to treat infections successfully, which were once life-threatening. Statement of the product: increase in the antibiotic resistant pathogens. Antibiotics are one of the most successful therapy in medicine. Their efficacy is being affected due to increasing number of resistant antimicrobial resistant pathogens. A study which was conducted in 2016 reported that yearly 700,000 deaths occur due to antimicrobial pathogens. And if nothing is done about this, then by 2050, it is estimated that about 10 million deaths occur annually. Another issue is that the drugs that we are currently using have specific biological toxicity. They disrupt nerve tissue cells, and like this is more harmful, especially for the children. For example, colicin, which is used for life-threatening infection, the risk of causing nerve kidney damage. And another is like tizicycline, which is used for the treatment of like notorious Acinetobacter. This antibiotic has the risk of causing liver damage. Another problem is that by now many of the pathogens have evolved into multi-drug resistance, MDR. We define MDR as a resistant to at least one agent, three or more microbial categories. The pathogens, they either go under mutation or they receive resistant genes from pathogens. Another issue is that we did not have enough new antibiotics developed. We all know it takes about 15 years to develop just new antibiotics, and like with the rate of increasing, with this rate of increasing resistant pathogens, it is not possible to deal with them. Like if there's this, if no new antibiotics are developed. The other issue is that the rate of approval for new drugs is really low. The approval for new drugs is tedious. It has to meet certain criteria and guidelines by the FDA, so it is not an easy task. So since most of the pathogens have are resistant to existing traditional antibiotics, it is urgent to develop an alternative to these antibiotics. So the objective of my today's presentation is to find an alternative to conventional antibiotics that target specific pathogens and antibiotics directly to the surface of specific cells. So now, with the advancement of nanoscience and nanotechnology, they have kind of provided a solution for this antimicrobial resistant thing. There are different nanomaterials that exhibit antimicrobial activity, such as metal ion, inorganic metals, or metal oxide. But these metal ions, they have biologic toxicity, so they cannot be used, and we need to find something else. So that is where carbon dots come. Like carbon dots, it was first discovered in 2004 by Zhu and her gang. They discovered it while they were purifying the nanotube. It is quartz spherical, which means it is not 100% spherical. it is fluorescent in nature. It is really small in size, 10 nanometer or below. It's a great chemical stability, means it cannot be degraded easily. It is soluble in water. It has the ability to absorb light and emit. It has low toxicity and has excellent biocompatibility, and also it can be like easily. The mechanisms, like how is carbon dots, like how do they microbial agents? There are like several mechanisms. One of them is in the absence of light or without photoexcitations. So in the absence of light, part of carbon dots has the antibacterial activity. The functional group on the surface of the carbon dots and the cell wall of the bacteria or microbes, they carry like opposite charges. So this creates electrostatic interactions, and because of this, they come together and form a strong binding. Now, because of this, carbon dots, what they do is they form a barrier, physical barrier around the bacteria or microbes, and isolate the bacteria from their surrounding environment. So this prevents the bacteria from like spreading nutrients and like maintaining their normal metabolism. So because of this, they are like it inhibits their growth and survival. So cities has the like since they can bind to the cell surfaces, they can inhibit the formation of films. Another is like disrupts cell membrane. So it causes cytoplasm leakage, apoptosis. The other thing about CDs is that it can bind to DNA and RNA once it's inside the cell, and this causes the denaturing of genetic material leading to cell death. As you can see in the pictures. The other mechanism is when there is light or a photon. In the presence of light, carbon dots, it act as a photosensitizers, and the process is called antimicrobial photodynamic therapy. This is a figure. It is a Jablonski diagram. It depicts the mechanism of antimicrobial photodynamic therapy. So this is a the blue colored. It is a photosensitizer, and when it receives light, it gets excited. So the excited electrons they move to the excited state, and when this excited states electrons come back down to the ground state, they release some energy in the form of heat or fluorescence. Now, at the same time, these excited electrons can also be converted to the T1 triplet state. So, when now this excited state in the T1 state come down to the ground state, they release energy in the form of heat or phosphorescence, and also generate ROS, which is reactive oxygen species. Now, in the same manner, carbon dots also generates reactive oxygen species, and we all know reactive oxygen species are very toxic to the microbes. So the mechanism is like the carbon dots, they bind to the cell surfaces of the microbes, and then like when it gets photoexcited, it gets excited and generates reactive oxygen species. This reactive oxygen species like do oxidative damages to the proteins, to the biological molecules. They like inhibit gene expressions, they damage the genetic material, and ultimately lead to cell death. Another mechanism in the antiviral is, so like we all know viruses need to like complete 4 of the stages. One is like attachment, conjugation, replication, and boarding to cause infection. So carbon dots can inhibit all these stages and prevent viral infection. A study was conducted in 2019. They treated Japanese encephalitis with CDs and then other DMA. And they introduced this to the host cell. And what they found out was that the viral title, a title which was treated with MCD was very low compared to the other one. Impact factors of CD's microbial activity, apart from the lights and all, CD's pollination antimicrobial activity. One is the size. Small carbon dots can easily pass through lipid bilayers, so they have more or better antibacterial activity compared to large size. Surface charge, the carbon dots are mostly positively charged and are mostly negatively charged. So negatively charged CDL activity. Surface functionalization, the core of the CD is carbon base and it is surrounded by inorganic molecules. And these molecules can be tailored according to the need of application, like including the antimicrobial activity.